Photosynthesis is a process that occurs in all green plants. The electron micrograph shows part of a chloroplast in a plant cell. The labeled starch grain in the chloroplast is 2.2 micrometers long. Calculate the width of this chloroplast between T and U. Right, so I'm going to have to find a paper, so I don't know how big this is going to be. Alright, so I'm measuring now on the actual paper. Measuring. Oh, that's perfect. That's 60 millimeters. Okay, so that distance is 60 millimeters. So that whatever we measure is usually the image. Okay, all right. The labeled starch grain in the chloroplast is 2.2 micrometers long. So 2.2 is the actual. Calculate the width of this chloroplast between T and U. The labeled starch grain. Okay. The labeled starch grain. So we're going to use the starch grain length to figure out T to U. All right. So we're going to have to start that again. I should have expected that. It's question 10 on an A-level paper. It wasn't going to be that easy. Okay, so then I'm going to measure the starch grain first of all. So if I figure out the measurement, what we're going to do is use the starch grain to figure out the magnification. And then once we've got the starch grain magnification, the magnification is going to be true for the other part as well. So if I'm looking at that, that is 26.5 millimeters. So starch grain is, the image of the starch grain is 26.5 millimeters. Okay, its actual is 2.2 micrometers long. Image divide actual. So 26.5 millimeters is, just checking, 26.5 times 1 times 2650. Okay, micrometers. So I get the units in the same place first, and then I divide 2650 by 2.2 to give me the magnification, and the magnification equals divide 2.2 is 1204.5. 1204.5 times. Okay, so if that's the magnification, now I just need to measure what T to U is. So T to U was equal to 60 millimeters, and so 60, let's just convert that into micrometers, equals 60, 60, that was too easy, 60,000 micrometers. So 60,000 micrometers divided by the magnification. Calculate the width of this chloroplast divided by the magnification. So image divided by magnification should equal actual is equal to 60,000 divided by 1204.5 equals 49.8 micrometers. Units are there. Okay, so yeah, that's how I would do that. Explain the relationship between the structure and functions of a granum in photosynthesis. 3 marks. Okay, so the granum, explain now. So the granum is composed of stacks of thylakoid membrane. Yeah, stacks of thylakoids, which are made of which are flattened disks, which are flattened disks of thylakoid membranes. So explain the relationship between the structure and functions, okay? So I think for every structural thing that I say, I want to say what role it has. So stacks of thylakoids, which are flattened disks of thylakoid membranes, this increases surface area, okay? This increases surface area for photosynthesis. Maybe too simplistic. Okay. Thylakoid membranes contain photosystems which contain chlorophyll to capture light for photosynthesis. I think that's basically it. Granum composes stacks of thylakoids. Explain the relationship between the structure and functions of a granum in photosynthesis. Increases the surface area to capture more light. Thylakoid membranes contain photosystems and electron transport chains to carry out the light-dependent stage of photosynthesis. Contain photosystems which contain chlorophyll to capture light. Thylakoids also have electron transport chains. Thylakoids also have electron transport chain for light dependent stage of photosynthesis. All right, well, I'm going for that. Structure and its role. That's my strategy. Don't know if I got all the marks. Explain the relationship between the structure, because sometimes they're a bit general about these things and sometimes they're oddly specific. So we'll go with that. Good morning. Right. Describe how starch is formed from the products of the light-independent reactions of photosynthesis. So, light-independent reactions, just going to speed up, of photosynthesis produce GP, or is it TP? Produce GP. GP is, so two molecules, so GP is a 3-carbon sugar, so 2 molecules of GP are combined to form glucose, alpha glucose, because we're talking about starch, okay? Glucose molecules are joined by condensation reaction forming glycosidic bonds Forming glycosidic bonds to bonds, okay, let's say to make a polymer or polysaccharide. Okay. Unbranched chains, unbranched coiled chains form the polysaccharide amylose. Branched chains which have 1 to 6, maybe I should have said 1,4 glycosidic bonds. Glycosidic bonds which have 1,6 glycosidic bonds in addition form amylopectin. Okay. Describe how starch is formed from the products of the light-independent reactions of photosynthesis. So we start from one of the intermediates in that cycle, and then we try and finish off with our knowledge of starch. So we just bridge those two things together. I think that's 4 marks. All right. Herbicides kill weeds by affecting their growth. The effect of herbicides on the production of starch in the leaves of Echinocloa cruskali. Hello. Hi. Yeah, I'll be there in a bit. Yeah, okay. Barnyard grass, should have just gone with that, has been investigated. The table shows the results of the effect of herbicides on the production of starch in the leaves. Okay, right, by affecting their growth. So presumably starch is needed for the growth. Okay, so if you affect the starch, then you can affect the growth, because starch can be used as an energy store. Or starch can be broken down to release glucose for respiration, which can then be used to provide energy for the growth of the plant. The table shows the results of 4 herbicides: diuron, propanil, linuron, and SWEP. Okay, so we've got the concentration increasing of each of these herbicides and the relative percentage of starch produced. Okay, so we see that as the concentration of these herbicides increase, we can see here that this is decreasing, this is decreasing, that's decreasing, and that's decreasing. Okay, though seems to be more to it than that, because right from the beginning we can see some of those are more effective. So that's gone quickly, that's gone less quickly, right, and that's gone the least quickly. Okay, it is thought that these herbicides act on the light-dependent reactions of photosynthesis. Devise an investigation that would produce quantitative data on the effectiveness of the herbicides on the light-dependent reactions of photosynthesis. Light-dependent reactions, a quantitative data on the effectiveness of herbicides on the light-dependent. Okay, so I think this is our Hill reaction core practical, because the Hill reaction is the light the Hill reaction is part of the light-dependent stage, and the core practical on photosynthesis measures this aspect of photosynthesis. Okay, so but we want to say that in simplified terms. We don't need to go into too much detail, but even once we've identified what the core practical is, we don't just want to fall into the trap of just describing the core practical. We do want to adapt it to be able to test these chemicals, these herbicides, to see what effect they have, what effect they have on the Hill reaction. Okay, so let's get into it. But let's just think about how we're going to do that first, and maybe I'll just leave my answer, because I'm running out of time a little bit. I might just leave my answer in a kind of planning stage rather than writing everything out. But what the basic reaction, remember, is that we isolate chloroplast. We isolate chloroplasts, and we might want to give at least one sentence of detail as to how we would do that, the precautions we would take to keep those chloroplasts intact, functioning, etc. So we isolate chloroplasts, and then we make some control solutions, but then we add DCPIP, DCPIP, we add that chemical, and then what we can do is measure the rate of decolorization. Because remember, adding DCPIP, and we probably want to explain, because it's a 6 marker, we probably want to explain what's the point of adding DCPIP. So DCPIP in the presence of light. So if light is there and DCPIP is there and chloroplasts are there, what should happen is light would result in the light-dependent stage happening, electrons to be flowing through the electron transport chain. Those electrons are used to reduce DCPIP, which makes DCPIP go colorless. Okay, so what we could do, how could we use this to measure the effect of these chemicals? So what we could do is we could have tubes where we've got different concentrations. So let's begin first with different herbicides added to chloroplasts. Well, would the herbicides act on the light dependent? Yeah, okay. So different herbicides added to tubes that contain chloroplasts, DCPIP, and our buffer. Okay, in the presence of light, and then in the absence of light as controls. Right, and then what we could do is at timed intervals we take samples out, because they've said quantitative data. So it's not just about looking at it and deciding whether the DCPIP has decolorized. At intervals, we need to take samples out, okay, and measure absorbance of light using a colorimeter. Okay, so we use the appropriate filter. So if the DCPIP is blue, right? If it's blue, then we want to use a filter on the opposite side of the color wheel, say orange, red, I believe. Okay, so we use an orange-red filter. We take samples out at intervals and we measure the absorbance of the colorimeter. Yeah, and essentially we after either a set time or and then we plot the absorbance versus time, and what we want to then compare, and in order to keep these comparisons valid, what we want to do is make sure that we keep certain biotic and abiotic variables the same. So in terms of biotic, what we would do is keep the species of the plant that we're getting these chloroplasts from the same. The volume, or the mass of plant material that we're using, we want to keep that the same. So those are our biotic variables. But what about our abiotic variables? So we probably want to keep the light intensity controlled in some way by using the same kind of bulbs, for example. We probably want to keep the temperature the same, because that can affect the rate of the light-dependent stage. Okay, so we're talking about our control variables, we're talking about how we're setting up, we're talking about what we're measuring, our dependent variables. And then finally, we will compare. And we're just going to say what analysis we're going to do. We're going to compare the time, or we're going to compare the absorbance, okay, the herbicide that lowers the absorbance the most has the greatest inhibitory effect on the light-dependent stage, because it slowed down the light-dependent stage, so it would slow down the decolorization of the DC pip the most. That's the approach I would take. I'll probably write that out a little bit more neatly. And that, I believe, is 2018 paper 1 all done. I will make a follow-up video to this, going through any major issues that I had in terms of marking and where my approach was wrong, where I would like to make improvements, etc. Thank you.